Hi friends, welcome back to my YouTube channel Learn Medicinal Chemistry Online. I hope you all are doing good. So in last video, I have explained alpha-1 receptor in detail, that is type of receptor, its mechanism, location, function, agonist and antagonist and their uses, and most importantly their structure. In today's video, I am going to explain alpha-2 receptors in detail. So let's begin guys. As I've mentioned earlier, all adrenergic receptors are G protein coupled receptors. There are four types of receptors in the body: G protein coupled receptor, enzyme linked receptor, ion channel receptor, and nuclear receptor, from which adrenergic receptors falls under G protein coupled receptor category because they have G protein in their structure. Okay. And G protein, I've also explained earlier, it's a 7-membrane helical structure which are spanning on the cell membrane. They have ligand binding site on the extracellular side, and they have G protein bound on the intracellular side. Okay, so here for alpha-2 receptor, the type is G protein coupled receptor, and type of G protein is GI. Okay, that is inhibitory protein. In alpha-1 it was GQ. Now it is GI, so obviously transduction mechanism will also be different as that of alpha-1 receptor. So transduction mechanism here is whenever any ligand binds with alpha-2 receptor, now that ligand can be adrenaline, noradrenaline or adrenergic alpha-2 agonist. Okay, when it binds with alpha-2 receptor, because the nature of this alpha-2 receptor is inhibitory, they will inhibit the enzyme adenylyl cyclase. Now, as I mentioned earlier, when a name ends with ASE in chemistry, it means an enzyme. So, adenylyl cyclase, the role of this enzyme is cyclization of ATP, that is adenosine triphosphate, okay, which is present in the cell. So, this adenosine triphosphate will be converted to cyclic adenosine monophosphate, that is CAMP, via this enzyme adenylyl cyclase. Now, because the alpha-2 receptor is inhibitory in nature, the ligand binding with alpha-2 receptor will inhibit this enzyme, so obviously decrease in the production of CAMP because the enzyme is now inhibited, so no more production of ATP to CAMP. Okay, now because there is less level of CAMP, that is cyclic adenosine monophosphate, in the cell, protein kinase will not be able to be activated by CAMP. Now what the CAMP was doing, cyclic adenosine monophosphate is responsible for activation of protein kinase enzyme. Now kinase means phosphorylation. So now because CAMP is not there in sufficient amount, I'm going to refer CAMP for cyclic adenosine monophosphate. So please take a note of that. Okay. Now this protein kinase will not be able to be activated by CAMP, and that's why there will not be phosphorylation of the cell, and which will lead to muscle relaxation. If that is a muscle cell, muscle relaxation will happen instead of muscle contraction. Muscle relaxation will happen. If it is a nerve cell will get inactivated. Okay, remember, because this is going to help you in learning function of this receptor. Okay, so first of all, inhibition of adenylyl cyclase, less production of CAMP, no activation of protein kinase, no phosphorylation, and that's why muscle relaxation. Okay, this is the transduction mechanism. Next comes is location and function. Now these alpha-2 receptors are present all over the body. Mainly, first is presynaptic axon. Okay, presynaptic axon. They are present in the presynaptic neuron. If you can recall the biosynthesis of noradrenaline, I've shown the reuptake mechanism. Okay, at that time alpha-2 receptor was present on presynaptic nerve also. So here also they are present in presynaptic neuron, where the role of this receptor is to decrease release of noradrenaline and adrenaline. How they decrease the release of noradrenaline? By negative feedback mechanism. I've explained earlier. Okay, I'll again explain. Negative feedback mechanism is when noradrenaline is present in sufficient amount. If it is synthesized in sufficient amount, then they will send signal to the brain that now you can stop the production of noradrenaline. So this mechanism is called as negative feedback mechanism. So excess of noradrenaline will bind back to the alpha receptor which are present on the presynaptic neuron, and that's why there the concentration of noradrenaline will be maintained in the synaptic cleft, that is the area between presynaptic and postsynaptic neuron. I've explained all this in biosynthesis lecture, so please refer to that lecture. Okay, next is spinal cord. Here these receptors produces analgesia. Liver cells decrease glucose level. Platelet aggregation. Smooth muscles of blood vessel, constriction. Okay, remember constriction. Skeletal muscle, they produce relaxation. Okay, as I've explained in the transduction mechanism, muscle relaxation. Ciliary body, decrease aqueous humor fluid production, and brain, increase sedation. Okay, let's repeat. Presynaptic neuron, they decrease the release of noradrenaline via negative feedback. Spinal cord, they produce analgesia. Liver cells, they decrease glucose level or production. Platelet aggregation. Smooth muscles of blood vessels, that is blood vessels, constriction. Skeletal muscles, relaxation. Ciliary body, now where the ciliary body is present, it is present in the eye. Okay, eyes. So here it decreases the aqueous humor fluid production. We'll see what it is in detail later on. Okay, aqueous humor production. Next, brain, increase sedation. Okay, if you can recall these functions, then you will be able to understand the uses of agonist. You will be able to correlate them. Okay, so next comes is agonist. The first agonist here is clonidine. Okay, now as for alpha-1 agonist, the series was zoline, and the examples was where xylometazoline, oxymetazoline, nefazoline, tolazoline. Here the series is nidine. Try to remember this series. Okay, that means the drug's name ends with nidine. So the first name here is clonidine. Now clo means chlorine has to be there, so can you see? Okay, chlorine is there. Then this part, what it is? It was present earlier also, imidazolina. Okay guys, I cannot repeat again and again because there are many structures to cover. So if you have any query regarding this heterocyclic structure, that is this one, you can refer to my second and third videos where I have explained all the heterocycles which are going to be used in third and final year. Okay, so this is imidazoline. If it were have a bond here also, then it would have become imidazole, but because there is no bond here, the structure is, this part is imidazoline, then this part is amino, and this part is dichloro, what it is? Benzene. Okay, now if you want to give numbering, the numbering will start from here. 1, 2, 3, 4, 5, 6. So basically it is 2 and 6 position contains chlorine, dichloro, 2,6-dichlorobenzene connected via amino to imidazoline group. Okay, and the name is clonidine. Next, now if you can remember clonidine structure, then apraclonidine should be easy for you. The only difference here is extra NH2, because the name is A, you can remember this way. A means extra amino group on which position? 4th position. See, can you see this amino group? That's why the name is apraclonidine. Remaining everything is same. So while drawing structures, you can draw clonidine first and then apraclonidine. So it will be easy for you. Okay. Next structure is brimonidine. Now because Bri is there, so obviously bromine is there. Can you see bromine here? Okay. Now this part is same. Imidazoline and amino. These is common. Here also it is there. Here also it is there. Imidazoline and amine is common. Next, what they have, which part is this? If you can recall again, this ring is queen oxaline. please refer to my second or third video. This ring is there. Okay, I've explained that. So this is quinoxaline. Okay, numbering of this ring will start from 7 and 8 because nitrogen is there, so obviously we'll give priority to nitrogen only. So it is 1 2 3 4 5 6 7 and 8. On 7th position this whole structure is attached, that is imidazoline and amino, and on 8th position Br is there. So if you can remember quinoxaline, then this substitution will be easy for you. Next structure is, so clonidine is done, apraclonidine is done, brimonidine is done. Next is dexmedetomidine. Do you know this drug? It's a very famous drug, and as a pharmacy student you should know it. Presidex. It's a very famous sedative agent, anti-anxiety agent, Presidex. So Presidex is a brand name. This contains this dexmedetomidine. Okay, now let's see what it is. Okay, here also this part is there, that is imidazoline is there. Instead of this NH, here it is CH-CH3, that is ethyl group. Okay, 2 carbon ethyl, and here it is what it is, this structure is orthotoluidine. My first video, basic aromatic compound. This is basic aromatic compound, orthotoluidine. Numbering will start from here, 1 2 3 4 5. Okay, ethyl and imidazolina. Okay, so see how easy it is. Dexmedetomidine. Okay, last structure in this category is, okay, now this doesn't fall under nidine series. The name is slightly different. It is guanbenz. Benz means, okay, benzene ring is there with 2 and 6 position contains what? Chlorine. So dichlorobenzene is there. Then what is this group? It is called as, now I am going to explain various functional groups while drawing structure. So simultaneously please focus on that also. This is guanidine. Basically what is guanidine? It is NH2 here, NH2 C double bond NH NH2. Now here only one hydrogen is there because the other hydrogen is replaced by, now what is this group? Imino. Okay, CNH2 is amino, CNH is imino. Amino, imino. Okay, try to remember the difference. Try to understand the difference. Okay, now here this part is guanidine. This part is guanidine. This part is amine. Now H is again replaced. Okay, valency is satisfied everywhere. CH double bond NH. See, it is CH. I'm so sorry, the structure is not so clear. Here it is CH double bond NH, and here it is 2,6-dichlorobenzene. The structure is of guanbenzene. So now can you understand why the name is guan? Because it contains guanidine and it contains benzene ring. That's why the name is guanbenza. Just you have to remember imino group. Okay, isn't it easy, guys? Next, guanfacine. Again guan is there, so can you see this group is guanidine. Okay, this group is CH2C double bond O is acetyl. It is CH3C double bond O, but one hydrogen is replaced here with this part. Okay, so CH2C double bond O acetyl, and rest of the structure is same. So if you want to compare these two structure, only this part is different. See, here it is imino, and here instead of imino acetyl group is there. Remaining everything is same in guanbenz and guanfacine. Next is methyl dopa. Okay, you can refer the structure from internet, no problem. Okay, it is easy. Just add methyl group to dopa structure, that is 3,4 dihydroxy phenylalanine. You will get this in biosynthesis, the video where I explained biosynthesis of noradrenaline. There the structure of dopa is there. Okay, just add methyl group to dopa. Still, if you have any query, I'll try to cover, I'll draw this structure in my next video. Okay, so methyl dopa, a very famous antihypertensive agent. Okay, so we've seen all the alpha-1 agonists. Now let's see the uses. Okay, first use is hypertension. Now you will wonder that while explaining location and function, I've already told you that alpha-2 receptor produces blood vessels constriction. So if it is producing constriction, how it is used in hypertension? Because in hypertension, the purpose is to reduce blood pressure by dilating the blood vessel. So here it does so by this mechanism. As I've told you that alpha-2 receptors are also present on presynaptic neuron, and there, see here, this function, decrease release of noradrenaline. So by this mechanism, it is used in treatment of hypertension, because what they do, they decrease the release of noradrenaline, and that's why they decrease blood pressure indirectly. So that's why it is used in hypertension, though they cause blood vessel constriction. Okay, next use is analgesia. As I've explained you that on spinal cord these receptors are present where they produce analgesia, sedation, sleepiness. In CNS, the action is sedation, brain is sedation. So that's why the agonist, alpha-2 agonists are used in sedation. Okay, they are very famous. As pre-anesthetic medication to relax the skeletal muscles. They are used. Okay, so you can add one more use, pre-anesthetic medications. Okay, also they are used as muscle relaxants. So there are total how many uses? One is hypertension, analgesia, sedation, then what I've explained, skeletal muscle relaxants, that is muscle relaxants, pre-anesthetic medication. Also they are used in ADHD. Now what is ADHD? Attention Deficit Hyperactivity Disorder. Now this is mainly seen in children, the children who are hyperactive or overactive, who cannot focus or concentrate on one thing. These agents are used because, as I've told you that okay, here I'm not explained, but as I told you that these agents are used to reduce anxiety, because in anxiety the system is hyperactive. So to relax the system, okay, to inhibit the system, that these agents are used. Okay, in ADHD, in children who are hyperactive, overactive, these agents will reduce the anxiety and will help them to focus on to concentrate. So they are also used in ADHD. Now there is one interesting use, that is glaucoma. Now what is glaucoma? It is blindness. It can be temporary, it can be permanent. Okay, now what happens in glaucoma? The optic nerve inside the eyes, optic nerve is present, which is responsible for, now this optic nerve, okay, my diagrammatic skills are not so good. These optic nerves are responsible for sending signal to the brain about the vision. Okay, let's say this is eye, so this is optic nerve. Okay, and this optic nerve gets damaged due to excessive pressure by aqueous humor. Now this aqueous humor is a liquid which is present inside the eye, and this is continuously removed, okay, by a normal mechanism. Will not go in detail because we are going to see that later in detail. So normally these aqueous humor is excreted and produced in balanced manner. But what happens, sometimes the aqueous humor formation increases, which leads to excessive pressure on this optic nerve, and this optic nerve gets damaged and there is no signal to the brain, and that's why loss of vision. What happens? Loss of vision. This loss of vision is due to damage of the optic nerve. This optic nerve is damaged by excessive pressure produced by aqueous humor. This aqueous humor is a liquid present in the eye. Okay. Now, how these agents are used to treat glaucoma? Because as I've explained that here they act on this ciliary body where they decrease the formation of this aqueous humor. So if this aqueous humor formation is decreased, very less aqueous humor will be produced and no more pressure will be exerted upon the optic nerve, and hence optic nerve will be safe. That's why they are used in glaucoma. Okay, so try to correlate the function and uses. It will be helpful for you. And next, last part is antagonist. Now there is no much research on this and they are not significant as very much, so I have not covered the structure. Example is yohimbine. Wherever I have not drawn the structure, you can refer it from the internet. Okay, now you know the basic heterocycles and aromatic compounds, also numberings, so you can draw it easily. Next, yohimbine, that is antagonist. There is only one antagonist, which is selective alpha-2 antagonist. It is used in the treatment of erectile dysfunction. Okay, so that's how it is done. Alpha-2 receptors are over. We have seen the type, we have seen the transduction mechanism, location and function. Now this part is very important because it will help you to understand the uses of agonist. Agonist is something which will produce same action as that of natural ligand. So whatever action noradrenaline is going to produce, the agonist will produce the same action. Okay, so all these actions will be produced by alpha-2 agonist, and that's why uses also will be same. Next, we have seen the agonist, that is Clenidine series, Clenidine, apraclonidine, brimonidine, dexmedetomidine, guanbens and guanfacine, methyl dopa. Uses we have seen. Also antagonist, example of antagonist is yohimbine. It is used in the treatment of erectile dysfunction. Okay. Okay, so guys, I hope you understood the alpha-2 receptors in detail, and if you like my video, then please share and subscribe and stay tuned for more updates. Okay, bye bye guys, see you later till next video comes.